Good morning all the students. I welcome you all to the class. As we discussed in the last class about spectroscopy and its types, now we'll move on to UV visible spectroscopic analysis. Here we are going to discuss about principle, instrumentation and applications of UV visible spectroscopy. Before moving on to UV visible spectroscopic analysis discussion, first we'll revise the last class. That is some introduction to spectroscopy. What is spectroscopy? Answer for the same. And what is EMR? What are wave properties? What are particle properties? And what are the types of spectroscopy? What are the contents we are going to discuss under UV visible spectroscopy is the introduction, principle and instrumentation, quantitative applications, pharmaceutical applications and spectrometric titrations. Coming to introduction, it was invented, UV visible spectroscopy was invented in July 1941 by Arnold Beckman. Main theory of UV visible spectroscopy is molecular absorption at valence electrons. Valence electron here indicates bonding and non-bonding electrons present in the molecule. And under types of spectroscopy itself we have discussed, that is UV visible spectroscopy is coming under molecular absorption spectroscopy. So absorption takes place by the molecule, whole molecule, at valence electrons. Valence electrons are bonding and non-bonding electrons. In the diagram it is depicted as bonding and non-bonding electrons. When it is absorbing UV visible radiation as electromagnetic radiation, it is going to excited state. And what are the bonding electrons? Sigma and pi electrons. Non-bonding electrons are n electrons, called as n electrons. When they are moving to excited state, that is mentioned with electron with star. Sigma star indicates sigma electrons at excited state. Pi star indicates pi electrons at excited state. And likewise n non-bonding electrons also will move to pi star, that is excited state, and its energy level is depicted here. It is the actual principle principle or theory behind UV visible spectroscopy, that is electronic transitions. We can say with the single word, electronic transitions. Electrons will be transferred from ground state to excited state after absorbing UV visible radiation. That is the concept here. Main application of UV visible spectroscopy is we can analyze chromophoric substances. We will discuss what is chromophoric substance in the coming slides. What is the main application? Analyzing chromophoric substances. Now we will discuss about principle and instrumentation of UV visible spectroscopy. The flowchart represents the steps involved in discussion on instrumentation. First is classification or types of instrument, then source of light, monochromators, sample cell, detectors, recorder and spectral study. One by one we will discuss in the coming slides. Now we will discuss about classification of UV visible spectrophotometer. UV visible spectrophotometer is divided into two types, a single beam instrument and double beam instrument. Single beam instrument is of two types, filters and spectrophotometers, where double beam of only spectrophotometers. Here single beam represents only single intensity of radiation. Only single path of radiation is passed to the sample. That means we can analyze only one sample at a time, either sample or reference. A single sample compartment will be there to hold a single sample, and it will be the instrument will be passing only single intensity of radiation. That represents single beam. Beam represents radiation. Single beam represents only single intensity of radiation, where double beam indicates will be having in the instrument double indicates two intensity of equal intensity of radiation will be passed to the sample. That means at a time we can measure two samples. One is of reference, another one is of sample what you are going to analyze. So two samples, that means multiple samples at a time we can measure by using double beam instrument. That is the difference between single beam and double beam. Single beam will be having single intensity of radiation, wherein double beam will be having two equal intensity of radiation to analyze multiple samples, where here only we can analyze single sample in single beam. Example for single beam of instruments are filter types and spectrophotometers. Filters represents, it is named based on using monochromators, that we'll discuss in the next slide. What is monochromators? Filter means it is a filter used to select appropriate radiation what we are going to analyze. So those type of instruments are called as filter types. Then spectrophotometers will be having, from this instrument we can derive the spectrum as report after analyzing it, and it will be having monochromator, grating monochromator as monochromator part, where in double beam instrument it will be having monochromator type, that is of spectrophotometer. So spectrophotometers are available with both single beam and double beam, where filters are available with only single beam. You can see the schematic representation of both single beam and double beam. On your left hand side you can find single beam instrumentation, on your right hand side you can find double beam instrumentation, where the parts given: source, entrance slit, dispersion device. For single beam I am saying source, entrance slit, dispersion device, exit slit. This from entrance to exit are called as monochromators, then sample, then detector. So single intensity, you see from the source to detector only single line is given, that is single intensity of radiation is passed. So it is called as single beam instrument. Coming to the double beam instrument on right hand side, that is source, monochromator, then it is going to the mirror device, mirroring system in the instrument, that is called as collimating system in the instrument, where it will be having reflecting surface and refracting surface for the radiation, where one beam will be refracted, that is it will be passed into that mirror, and one will be reflected, that is coming back from the mirror, and it is going to two samples. So it is beam splitter that splits to equal intensity of radiation, that is derived from the source. Two equal intensity of radiations are divided at the beam splitter. That is beam splitter is nothing but a mirror. It will be having two surfaces. One surface will be passing transparent to the radiation, another one will be reflecting the radiation. Thereby the radiation is divided into two beams, that is of equal intensity. If one beam is having 50% intensity, another one also will be having 50% intensity. So both the radiations are going to two different sample cells. One is of reference, another one is of sample, and it is going to detector. Then the detected signal will be recorded as spectrum. That is the double beam spectrophotometer. Hope you have understood what is single beam and double beam instrument. In our laboratory we are having double beam UV visible spectrophotometer. So it functions similar to the schematic representation given for the double beam instrument. Coming to UV visible spectrophotometer, the main part of instrument that we are going to discuss about. If you take any spectrophotometer or any spectrometer, it will be having 5 common parts. What are the 5 parts any instrument will be, any spectroscopic instrument will be having means: source of light, monochromator, sample cell, and detector. What are the parts? Source of light for source for electromagnetic radiation. Then monochromator, it will be selecting means whatever wavelength we want. That is the role of monochromator. It will be selecting radiation from source of light. Then sample cell will be having sample where the radiation is passed into it to analyze the sample. Then detector, whatever radiation comes after absorption or emission from the sample to the detector, it will be detecting it. And then recorder will be recording the signal that is detected by the detector at last we will be getting spectrum. This is the common principle, working principle of any spectrophotometer. Now we will discuss what are the parts present in the UV visible spectrophotometer and how it functions. Firstly, source of light for UV visible radiation. How we will get UV visible radiation? For UV radiation, the source is D2 lamp. It is just a lamp which is made with D2. It will be having the filament that is made with D2. D2 is nothing but deuterium. Deuterium is the isotope of hydrogen H2. That is D2, here mentioned as D2. Deuterium lamp. It will be emitting radiation between the range of UV, that is 200 to 400 nanometer. In this range, UV samples will be absorbing UV radiation. Then for visible W, that is tungsten lamp. W represents tungsten filament. It is made with tungsten filament. Range of visible radiation is from 400 to 800 nanometer. That starts from UV range and ends before IR range actually in the electromagnetic spectrum. We have seen about electromagnetic spectrum in the last class. You just go through the electromagnetic spectrum to find out where UV and visible radiation are exactly lies in the spectrum, and it is measured by wavelength, by means of wavelength. Waves are measured by means of wavelength, that is represented as lambda. So lambda value is 200 to 400 nanometer and 400 to 800 nanometer. You can observe the D2 and W lamp in the picture, how it appears, and what is the mechanism, how it gives radiation means when it is electrically heated. That means when we are giving electric energy to the lamp, it will be converting it into radiation energy. So when we pass electrical energy to the D2 lamp, it will be and that means hydrogen present in the lamp will be electrically heated, thereby it goes to excited state, and to come to ground state it emits radiation. So to come to normal position, that is ground state, the excited hydrogen in the form of deuterium, it emits UV radiation. That is the concept. Likewise, tungsten lamp also, when it is electrically heated, it will be, after absorbing electrical energy by tungsten, it is going to excited state, and to come to ground state it emits radiation, that is in the form of visible radiation between the required range. So main mechanism of source of light is it converts electrical energy to radiation energy. Next, coming to monochromators as the part of UV visible spectrophotometer. What is monochromators? Mono represents single, chrome represents color, single colored light. Here we are discussing about light, that is EMR, electromagnetic radiation. Light here represents electromagnetic radiation. So monochromators are nothing but it will be selecting single colored light. Colored, so here represents wavelength. Single wavelength light is going to be separated from multiple wavelength light. That means multiple wavelengths are called as polychromatic light. It converts polychromatic light to monochromatic light. That is represented in the picture. The main principle of monochromators are dispersion. Dispersion means it is separating wavelengths from multiple wavelengths to single wavelengths. That is the role of monochromators. So from the source, whatever radiation is coming out is converted to monochromatic light. That is the light we want to analyze the sample, we want to use for analyzing the. Next, coming to the types of monochromators, they are divided into two types based on using instruments, single beam and double beam. If you are using single beam, what is the type of monochromator used? If you use double beam, what is the type of monochromator used in that instrument? For single beam, they are called as filters. For double beam, it is of two types, again prisms and monochromators. Coming to filters, what is the principle of this monochromators? Different types of monochromators are filters function like a filtering process by means of filtering of the radiation. That means it filters off unwanted radiation, only it selects the wanted radiation and it sends to the sample. You can observe in the picture, that is green interference filter, it filters off only green light and it sends to the sample. And next, coming to the prisms, you can find the picture on the top of the slide where prism functions like a dispersion process. It disperses the light what it receives from the source. Dispersed light, colored light represents dispersed light. light into individual wavelengths, where white light comprises of all multiple wavelengths, that is polychromatic light. White light represents polychromatic light, where colored light represents dispersed light or monochromatic light. So prisms functions like dispersion process. Coming to monochromators, monochromators functions based on diffraction process. Example of monochromator is grating monochromator. It functions based on diffraction. You can find in the picture monochromators have gratings or rulings on the surface where white light is passed and it is diffracting the radiation, and it is segregated into individual wavelengths. And this monochromatic light between the range from 200 to 800 nanometer of UV visible range is passed to the sample. What are the types of filters, prisms and monochromators are? For filters, absorption filter and emission filter, prisms, quartz and glass. Quartz used for UV instruments, glass for visible instruments, where monochromators are of grating. Monochromators are used widely. Next part of the instrument is sample cell. It is called as cuvette, and there are two types of cuvettes available for a UV visible spectrophotometer. It is cylindrical shaped cuvettes and rectangular shaped based on its shape. Cylindrical cuvettes like test tubes, they appears like test tubes. They are used in filter types of instruments. Example is calorimeter. You should have used cylindrical cuvettes there. And rectangular shaped cuvettes you can find in double beam spectrophotometers, where UV visible spectrophotometer uses rectangular shaped cuvettes. For calorimeter, cylindrical, UV visible, rectangular, single beam, cylindrical, double beam, rectangular cuvettes. Coming to rectangular cuvettes, for UV analysis, we have to use quartz coated cuvettes, rectangular shaped quartz coated cuvettes. Why we need to use quartz coated cuvettes for UV analysis means glass will be absorbing UV radiation. So to avoid loss of radiation by the cuvette, we need to use quartz material. Quartz is translucent to UV radiation. That means it will not absorb UV radiation. Hence it is coated with other two sides, you should have seen in the cuvette, two sides are having rough surface, that is coated with quartz material. Where visible analysis, we can use quartz or glass cuvettes we can use, no need to have the coating. And since visible light will not be absorbed by the glass, it is transparent to the radiation. So for calorimetric analysis, we can use glass cuvettes. For UV analysis, that is from 200 to 400 nanometer, we must use quartz cuvette to avoid the loss of radiation. This is how cuvettes are selected, based on instruments and based on radiation it is selected. Capacity of cuvette normally 3.5 ml and path length 1 centimeter. It is important, path length to note, you have to note it down, since this path length is applicable for quantitative analysis, and to obey Beer-Lambert's law, we must use 1 centimeter path length cuvettes. Then after choosing the sample cell, we need to select the sample. What type of sample, what type of solvent it is used to prepare it, what is the concentration range we need to use for analyzing it by using UV visible spectroscopy. First we'll see the nature of sample used for UV and visible analysis. For UV analysis, we'll be analyzing colorless samples. For UV range from 200 to 400, we'll be analyzing colorless samples. Where at visible indicates we can differentiate the colors. All Vibgyor colors can be differentiated, so it is called as colorimetric analysis also. So colored solutions, color, visible indicates color, colored solutions are measured by using visible range, that is from 400 to 800 nanometer, where UV wavelength is having more energy comparing with visible radiation. You have to compare the energy of radiation with the wavelength. When wavelength is inversely proportional to energy, as we have discussed in the last class. Energy is indirectly proportional to wavelength. When wavelength increases, energy decreases. And what type of samples are chosen for analysis by UV visible? Chromophoric substances. I discussed in the beginning itself. Chromophoric substances of any type of sample should be chosen. What type of solvents are used to prepare solution? Means polar solvents. Mostly water, methanol and ethanol. Polar solvents are used. So your sample must be in polar in nature to analyze by UV visible method. And then concentration range. What concentration range we can analyze? Means microgram to milligram samples. Microgram to milligram samples. So what type of sample finally we need to analyze by UV and visible method? Monophasic. It is not biphasic solution. Monophasic diluted solutions of chromophoric substances, either colorless or colored. Monophasic diluted solutions. Diluted solution. Here dilution is important. If you are taking any solid material, you have to use proper polar solvents, and then we need to dilute it to get the concentration range from microgram to milligram to analyze it before. And once the sample is ready, you will be keeping the sample in the sample cell and you pass the radiation from monochromator. Actually radiation comes from source and it passed to monochromator. Now it is ready to pass to the sample cell. After passing it, what type of interaction takes place between sample and radiation? So chromophores present in the sample will react with UV or visible radiation, thereby absorption of photons by the sample by chromophores occur. Then electronic transitions takes place, that is sigma electrons, pi electrons and non-bonding electrons from ground state, they will be moving to excited state, and we need to analyze, the instrument will analyze how much amount of radiation is absorbed by these electrons to move from ground state to excited state, thereby you will get absorption spectrum. So absorption spectrum will give about how much amount of radiation is absorbed maximum by the chromophore. Now let's move on to the most significant part of UV visible spectroscopy, that is chromophores, oxochromes and types of bands appear due to it. What is the compound or group responsible for absorbing UV visible radiation is chromophore. If you take any molecule, it must be having chromophoric system. So now we have to know what is chromophore. What is the role of it? It is nothing but a conjugated system to absorb electromagnetic radiation. What is conjugated system? It is nothing but a double bonded or unsaturated system present in the molecule. How double bond is formed in the molecule? It is due to electrons, sigma and pi electrons. More reactive electron or more reactive bond here is pi electron or pi bond. The bond due to pi electrons. So pi electrons are responsible then to absorb the radiation, UV visible radiation that is present in double bond. What is conjugated double bond? It will be having alternative double bonds in the molecule. You should have found in the aromatic ring, that is having alternative double bond, that is conjugated system. So definitely it will be having UV visible absorption. Minimum a single conjugated system must be there in the molecule to absorb UV visible radiation. And what is the oxochrome? What is the role of it? It will not directly absorb UV visible radiation, but it has influence on chromophore. What influence it has? It is extending the conjugation, or it is extending the chromophoric nature in the molecule. How it will extend? What is the role of it? It will be donating its electron present in it, so that there will be a group of compounds of electron donors. So the electron donating groups are called as oxochromes, and these groups will be donating its electrons when it is attached with a chromophoric system in the molecule. It will be donating its electron to that chromophore or pi bond, a double bond, so that it will be extending its conjugation. Thereby that particular chromophore requires less energy of absorption. When energy is less, it will be having more wavelengths, so it will be shifted to higher wavelengths or longer wavelengths, that is towards visible range. That is the role of oxochromes. So oxochromes reduces absorption energy for the chromophore. How means by extending its conjugation. Now we'll see what are the types of chromophores and oxochromes. There are two types types of chromophores, dependent chromophores and independent chromophores. Dependent means it will be depending some other chromophore for imparting the color or for giving the color. Independent chromophore means they themselves they will be having color of the compound. Example is azo and nitroso. These compounds will be having color on its own, where ethylene and acetylene will have to depend on some other chromophores to impart color. Now we'll see what type of electrons they will be having and what is the electronic transition. Pi electrons, dependent chromophores will be having pi electrons and the transition from pi to pi star, where independent chromophores will be having pi electrons and non-bonding electrons both. So the transition from pi to pi star and n to pi star. And what are the bands appear due to this transition in the UV visible range? B band, E band, K band and R band. What is B band? B band is due to benzenoid ring. Benzenoid represents aromatic or heteroaromatic group present in the molecule, and E represents ethylenic group in the molecule. So band due to ethylenic group will give E band, and K band is due to conjugate group. R band is due to radical group. So R represents radical group, where K represents a conjugate group. So next we'll see the types of oxochromes. There are two types, acidic and basic. Example of it, OH, OR and NHR. They will be electron donating groups, will be active in acidic solution, where basic, next one is basic, nitrous oxide, carbon monoxide and CN. Nitriles will be active or will be donating electrons in basic solutions. So they are of two types, acidic and basic oxochromes. So these are the types of chromophores and oxochromes, and what are their roles and what are the types of bands. Now we'll see the important practical part when you choose solvent for UV visible analysis. When you use less polar solvents, the transition will require higher energy. You can see the energy level and solvent polarity in the x-axis, y-axis energy level, and the transition of electrons, pi electrons and n electrons from ground state to excited state. At less polarity, it is having high energy to go from ground state to excited state, where at more polar the energy of transition is reduced. That means it has higher or longer wavelength. Energy of UV visible radiation is reduced for its absorption, and it is soon it is going to excited state, from ground state to excited state, pi to pi star and n to pi star. What happens when polarity is high or when you use more polar solvents? They forms with the compound, solvent forms with the compound intermolecular hydrogen bonding. Thereby it stabilizes conjugation nature. When conjugation nature is stabilized and it is extended, definitely it moves to longer wavelength region. That means it reduces energy of absorption when conjugation is extended. That is what here the solvent effect is shown in the picture. You can observe the picture for solvent polarity and its effect on energy of UV visible radiation absorption. Next, coming to the different types of shifts occur during the UV visible analysis when we get the spectrum. There are four types of shifts occur in the UV visible spectrum. First one is blue shift, then red shift, hyperchromic shift and hypochromic shift. And the x-axis represents lambda, that is wavelength, radiation range. What is the range of radiation for UV visible? 200 to 800 nanometer. And in the y-axis it shows absorbance. And the spectrum shown in the green color, where blue arrow marks are showing different shifts from the peak appears. From 200 to 800 in the x-axis. Note down 200 to 800 is the range of UV visible range. When the lambda max, you can find in the x-axis lambda max, it is the important parameter, shows qualitative aspect of the molecule. that represents maximum absorbance at particular wavelength. What is lambda max? Maximum absorbance for a molecule at particular wavelength. It will be shifted towards 200 means that is blue shift. If the lambda max is shifted towards 800 nanometer, then it is red shift. Blue shift will be having less wavelength and higher energy, where red shift will be having longer wavelength and lower energy, where hyperchromic shift will be having high intensity color, hypochromic shift will be having less intensity color. If the peak moves up, then it is hyperchromic. Hyper means high colored compound. Hypochromic means less colored compound, which is giving the lambda max. But in the lambda max no change occurs, but the intensity of radiation changes with the hyperchromic and hypochromic, where lambda max is shifted with the blue and red shift. Blue shift is otherwise called as hypsochromic shift, where red shift is otherwise called as bathochromic shift. Hypsochromic, bathochromic shift. And the most important part of UV visible spectrophotometer is detectors. What is the role of detector? Detector represents, the name itself it represents, it detects something. So what it detects in the spectrophotometer? It detects radiation. So how much amount of radiation it receives? It is converting that amount of radiation into electrical signal. So equal amount of electrons will be produced based on the photons it receives. It is the role of detector. That means it converts photons to electrons. By what means photons and photons are converted to electrons? What detector has to convert it? It has electrodes. What type of electrodes? Cathode and anode. What are the two types of electrodes? Cathode and anode. Negative electrode and positive electrode respectively. What is the use of negative electrode? It receives photons. H nu equal to H nu, that is radiation energy. Already we discussed in the last class. What is H nu? Is a photon or a single photon, that is equal to energy of radiation. It is received by cathode, and this cathode emits equal amount of electrons. Electrons are negatively charged, so it will be moving towards positive electrode, that is anode. When anode receives the electrons, it passes means current. It passes current in the electric circuit. Thereby, direct current will be converted to alternating current in the circuit. Direct current DC is converted to AC, and thereby electrical signal is produced, that is proportional to the radiation energy what cathode received from the sample. And then this electrical signal then again passed to the recorder. There you will get your spectrum recorded in the recorder, so that you will be getting spectrum. That spectrum will be saying about how much concentration of sample was there in your prepared solution, diluted solution. So the solution will be having absorbing species, that is chromophore, that would have absorbed the radiation. After absorption, some amount of radiation is received by the cathode. This amount of radiation, intensity of radiation, is converted to electrical energy by means of detectors. Then electrical signal is passed to the recorder to record the spectrum. So whatever report you are getting from the spectrophotometer is called as spectrum. We cannot say it as graph. It appears like graph, but we cannot say it as graph. It is called as spectrum. If you have single spectrum, then it is called as spectrum. If you have multiple reports, then you can call it as spectra. More than one spectrum if you receive, then that is called as spectra. Spectra is plural form, spectrum is singular form. And likewise, if you see the picture of all the detectors, each detector will be having a cathode and anode, where cathode receives radiation and it emits electron. These electrons are absorbed by anode and it passes electric signal in the circuit. What is the role of photomultiplier tubes, and where you will be finding, and what type of instrument you will be finding these detectors means, in filter type of instruments you will be finding photovoltaic cells and phototubes as detectors. In spectrophotometers we will be finding photomultiplier tubes and photodiode arrays. What is the use of photomultiplier tubes? It is most important one. You can see the orange colored mirrors, they are nothing but anode, dinodes, it is called as dinodes, where cathode receives radiation and it passes to the anode, where one you see the first dynode, it receives only one signal and again it reflects the radiation to another dynode. Dinodes are nothing but multiple number of anodes. Anodes are arranged opposite to each other, so when the reflected radiation goes to another anode, it is multiplied again twice. Likewise, the signal is multiplied many times, so that even weak signal, if you receive from the sample, that can be multiplied and analyzed by the detector. So sensitivity of photomultiplier tubes are high comparing with other detectors like photovoltaic cells and phototubes. Phototubes and photovoltaic cells will be having less sensitive than photomultiplier tubes and photodiode arrays. Diode array also functions with a grating monochromator, where diffraction occurs, thereby whatever radiation we are receiving from the sample is splitted into individual wavelengths, so no wavelength is neglected without detecting, and each signal, even from weak signal to strong signal, all the signals, electric signals, radiation that is received from the sample is detected by photodiode arrays also. Where we are using grating monochromators, here we are using, in photomultiplier tubes we are using dinodes. These are the functions of detectors and what instruments you can find these detectors and what is the mechanism. Here now we have discussed. And the last part of instrument is the recorder. The role of recorder is it draws electrical signal into spectrum. What are recorders? They are nothing but analog meters. They will be having digital display or LCD. So they will record electrical signal into spectrum, and we can take the printout of this spectrum and we can use for quantitative analysis, qualitative and quantitative analysis. Next we will see about spectral study. After getting spectrum from recorder, we can analyze the spectrum by means of qualitative parameter and quantitative parameter. There are two types of analysis you know already, that is qualitative analysis and quantitative analysis. What is the qualitative analysis? What is the qualitative parameter in UV visible spectroscopic analysis is Lambda Max. Lambda Max is called as qualitative parameter, where how we can analyze it means taking the spectrum, wavelength versus absorbance, wavelength in the x-axis and absorbance in the y-axis. You can find the peak in the spectrum, and that is at 525 nanometer. That particular nanometer wavelength, nanometer is nothing but here wavelength, that is called as lambda max, maximum absorbance for a particular molecule at a particular wavelength, that is called lambda max. So that is the qualitative parameter. So we can analyze what molecule it is. When comparing this value lambda max with a standard value, we can identify what type of molecule it is. So it is called as qualitative parameter. What is the qualitative parameter for UV visible analysis? Lambda max, that you need to remember. Next is quantitative analysis. What are the quantitative parameters? Concentration versus absorbance. If we know the concentration only, we can quantify the sample. So the main parameter is concentration, that is taken in x-axis, where in y-axis absorbance is taken. Optimum absorbance value here is 0 to that is 0.1 to 0.9. 0.1 to 0.9 is the range of absorbance, optimum absorbance in absorbance units. Where absorbance has no unit, it is said with absorbance unit. And x-axis concentration. So you will be plotting calibration curve as shown in the figure, and from which we can analyze sample concentration. Once you plot the linear curve, then from which we can get the sample curve by means of extrapolation. So this method is widely used to quantify the sample. So what is the quantitative analysis parameter? Quantitative parameter, concentration versus absorbance. Concentration in x-axis, absorbance in y-axis. Through calibration curve we could find sample concentration. Thank you all for patient listening, and stay safe, stay at home. Thank you.